So hello, everyone. Thank you for having me here. Quite exciting to talk about this. So the investigation of protein structure is important, which gives insight into the function of proteins, which is then used for various drug discovery, for understanding of disease mechanisms. And the first step in the investigation of the structure is the assignment of the backbone. And the backbone is the primary structure, which consists of sequentially connected amino acids through peptide bonds. And these assignments are usually done through sets of triple resonance experiments, where in the triple resonance experiments, the magnetization is transferred from the hydrogens to the carbons to the nitrogens, which give the three-dimensional spectrum, which then the amino acids in the backbone will be seen as signals and peaks. And within the spectra, the signals could be sequentially connected, and their chemical shift positions are then used for the assignment of the amino acid type. So on the right here, you can see an example of this three-dimensional spectrum on the hydrogen, nitrogen, and the carbon axes, where the red dots would be the signals from the amino acids in the backbone. And usually they're separated into strips, which the strips can then be sequentially connected. And on the left here, you can see the HNCA experiments, where the experiments are named after the magnetization transfer. And in the HNCA experiment, the magnetization is going from the hydrogen to a nitrogen and to the C alphas. But the C alpha magnetization transfer can go both to its own C alpha and its preceding one, and hence you get two signals within the hydrogen dimensions. And within the pairs of signals, usually you do a second set of experiments, which is the HNCA experiment. And within this experiment, you get a signal only from the preceding C alpha. And so in software like CCPN, if you overlay the experiments, you can identify which is the main and the preceding signal. And when you do this for the strips of all the signals, you can sequentially connect them together. So on the top left, you see an example of the strips for different signals. And hence, by traveling along the I and I-1 signals, and then traveling along the carbon dimensions, you can sequentially connect the signals into long chains. And the long chains can then be assigned back to the primary sequence. And on the bottom left example, you can just see the pairs on the 3D dimensional figure, where the green box is the pair of signals, and in the red line, you can see where you're traveling along the carbon dimension to find the correct sequential pair. And usually you also do the C beta C alpha experiments to get the information on the C beta chemical shifts. And the C beta chemical shifts, including the C alphas and the hydrogen and nitrogen positions, can be used for the assignment of the amino acid type. And the HNCA is the most sensitive experiment, and however, it doesn't contain the information for the C beta chemical shifts. And the C beta chemical shift is usually used for the assignment of the amino acid type. So as you can see, the various distributions of where the chemical shifts can go for the C beta are quite different for the amino acids. For the C alpha, they're similar, except for glycine, where glycine is quite separate from them. And hence, once you have the chemical shift for all the signals, you can use them to create, to assign a probability vector for the signal belonging to each amino acid types. However, this becomes more difficult for larger proteins because of degeneracy issues. So for example, here you can see the HSQC for an 8 kilodalton protein and 80 kilodalton protein. And for larger protein, the resolution is worse. And on top of this, because there are more amino acids in the backbone, there is significant overlap of the signals. And hence, trying to do the sequential matching can become very difficult because the signals overlap and there will be several potential matches for each sequential assignment. And so in the example here, you can see that we're trying to assign a sequential match for tyrosine 190. And there are 3 potential matches. And it will be very difficult to distinguish which will be the correct one. So tryptophan 191 is the correct one, but tyrosine 40 and aspartic acid 41 are also good sequential matches, and hence it is difficult to assign the correct sequential connection. And one of the ways to overcome this is to increase the resolution. But even if you increase the resolution of your spectra, there is inherent coupling between the C alpha and the C beta carbons in the proteins, which will be seen as splitting in the signal. And you will see these doublets within the spectra at 35 Hertz. And these doublets are reducing your resolution in the assignment of the chemical shift and the sequential assignments. And there are various ways to overcome this. And one way to overcome this was proposed by a team in Harvard, led by Haribabu Alfanari and Scott Robson. And their idea was to use isotopically labeled proteins. So if the bacteria which are expressed, the proteins which are expressed in the bacterium and the bacterium are grown in isotopically labeled pyruvates, which can be labeled with carbon-13 in either carbon 1, 2, or 3 positions, the proteins will be also isotopically labeled from the pyruvate. And their idea was to assign the entire backbone for large proteins with single HNCA experiments without touching the C-beta chemical shifts. And the way they did was they grew the bacteria in a mixture of 2 and 3 labeled pyruvates. And so through various synthesis of the different amino acids, the amino acids can take different paths. And depending if the 2 or 3 pyruvate is used as a precursor, you get amino acid dependent line shapes, where either the C alpha beta coupling is overcome, so you get a strong central peak, or you can get you can see like example of shoulder peaks here. So they conducted this experiments with MBP and GB1. So MBP is a maltose binding protein. And on the left here, you can see the example of the HNCA spectra for the different amino acids. So you can clearly see that for alanine 188 at the top left corner, you have quite a strong central peak. But for valine 181 at the bottom corner, you get a strong central peak and these smaller shoulder peaks. And hence, for the experimental labeling of the carbons, they were able to generate amino acid dependent line shapes within their HNCA spectrum. And after they did this experiment for various proteins, they were able to, they parameterized these peaks. So they parameterized them as having a strong central peak and weaker shoulder peaks. And for each amino acids, you could parameterize them by the chemical shifts, which is the hydrogens, nitrogens, and then the C alpha. And on top of this, they measured experimentally the coupled to uncoupled peak ratio between the central peak and the shoulder peaks. So they would measure the spectra for MBP. They knew what the correct labels were for the amino acids. Then they would experimentally measure the coupled to uncoupled peak ratio for each amino acid type, and hence they could give distributions for where these amino acids should fall onto. And on top of this, they would also parameterize the J coupling distance between them, which was also amino acid dependent. And so by measuring these distances, they knew the distributions that these amino acids should fall into. And this gives extra information for sequential assignments and gives also extra information on the assignment of the amino acid type. So for example, here, when we're trying to do sequential fitting for valine 259, we get potential sequential matches with isoleucine 212 and glycine 260, where both of them can fit well in the carbon dimension. We're trying to sequentially match them. But now you can break this degeneracy by looking at the line shapes. So in the green, you can see the line shape of isoleucine 212, which doesn't fit well with valine 259. However, glycine 260 has a very good sequential match on the red. And in this way, you could break the degeneracy and have quite a confident assignment in the correct sequential match. And on top of this, by looking at the correlation coefficients, could give a confidence in your sequential assignment. So the correlation coefficient for glycine 260 is 0.982, which is really high. And hence, even visually and with the correlation coefficient, you could quite confidently assign that glycine 260 was the correct peak as a sequential assignment to valine 259. And this way, with single HNA spectra, they were able to assign 85% of the MBP protein. So in the red here, you can see the uniquely assigned long chains. In the yellow are the residues which were seen, but not sequentially connected, but they tried to assign them anyway, but were assigned incorrectly. And in black are the residues which were not seen in the HNCA spectrum. And so with one experiment, they could assign about 45 or 42 long chains, which then they could relate back to a primary sequence and assign 85% of the protein without touching the C-beta chemical shifts with only a single HNA experiment. But this process can be quite difficult. quite laborious. So they had to write specific software to do a line shape matching. And on top of this, even when you have the long chains of the sequential assignments, trying to relate them back to a primary sequence and assign amino acid type, by visually looking at the line shape can be quite difficult because you have to look at different combinations and you have to try to assign them yourself. And so we thought we could automate this process with neural networks, because fundamentally, when you take images of the spectra of the each peaks, you will see, so for example, on the right here, you can see an example of an image in GB1. In the yellow, you see the quite strong central peak, and then you can also see the shoulder peaks. And this is the line shape which they could experimentally show you for the polygroup labeling. And trying to assign amino acid type for these images is fundamentally image classification, which are done really well by neural networks. And so our idea was to train a neural network, which is given an image of a signal from a HNCA spectra, could give a probability on assignment for each amino acid type. And these images would also be supplemented with the chemical shifts. So now the neural network would give a prediction, not just for the chemical shifts, but these images from the signals of the experimentally measured HNA spectra. And to do this, we had to generate a large training database, where the training database consisted of synthetically generated three-dimensional spectra of the labeled pyruvate HNA spectra. So we had, for the training data, we had a signal image, which was our synthetic generation of the spectra, supplied with the chemical shift positions in the first row of the. So we supplied the chemical shifts in the first column, supplied the hydrogen chemical shifts in the last row, and in the bottom corner we had the nitrogen position. And on top of this, we had the signal image. So the training data consisted of the signal image, the chemical shift, and the ground truth probability of what the correct assignment should be. So the process consists of randomly choosing an amino acid type. Once we had a random amino acid type, we chose a chemical shift from the distributions from the BMRV database. So in here, you can see these 5 distributions. The chemical shifts were from the BMRV database, while the coupled to uncoupled ratio between the heights and the J coupling, which were measured experimentally by the team in Harvard. And so when we chose an amino acid type randomly, we picked these values from their known distributions. And once we had them, we assumed Lorentzian line functions in all the 3 dimensions. So we had a Lorentzian in the carbon dimension. So we had a Lorentzian in the nitrogen dimension, a Lorentzian in the hydrogen dimension. And the carbon dimension, we assumed a strong central Lorentzian with 2 smaller, weak shoulder Lorentzians. And these, the heights between the Lorentzians, between the shoulder ones for the carbon dimension were from the known distributions. And the J coupling was also from the known distributions of the measured pyruvate spectra. And afterwards, we generated these synthetic 3-dimensional spectra. So within the spectra, we had our generated signals. And for each signal, we knew what the parameters we used to generate them. And hence, we could assign the ground truth of what their prediction should be. Because we know the parameters which were generated for the chemical shifts and also the J coupling and the coupling to uncoupled fractions from the distributions, we could assign a probability vector belonging to each amino acid type. So you can, with the line shape matching, you can quite confidently eliminate some amino acid types. So on the last image, on the 6th one, you can see the overlap of their distributions. So some amino acid types have quite a lot of overlap, so it'd be difficult to distinguish between them, but we could distinguish quite a lot of them from each other. And so for each signal, after we generated the 3-dimensional spectra, we also added various experimentally measured distortions. So for each signal, we added phase distortions, we added random noise, we added baseline, we simulated the entire process the experiment was measured. So we added the truncations and window functions and all the various Fourier transforms. And hence, we could generate tens of millions of these synthetic images with the corresponding ground truths. So on the right here, you can see an example of this. So we have an example of the synthetic image for a signal, which was the input to a neural network. And on the bottom, in the blue, you can see the predictions from the neural network, and the reds were the actual correct predictions from the 5 parameters which you used to generate this image. And so after training on tens of millions of images, we could use this neural network and attempted to do an assignment on an experimental spectrum. But we also added other distortions. So we also, for each image, we added random shifts to simulate the user not essentially picking each peak, which made the neural network more resilient. And here you can see the architecture example. So we had 4 hidden layers with all fully connected networks, and then the input image is the image is the input to a neural network, and the outputs are the probability vectors. And after we tested this on the experimental spectra of MVP and GB1, so we knew what the images should be. We had the images, the experimental images from the spectra. We knew what the correct assignment was. So we wanted to see how accurately could the neural network predict this. And for each individual residues, we could predict 56% of MVP correctly in the first position. So the prediction would give probability type for each amino acid type, but if you chose a top probability, the first prediction was correct 56% of the time. And for MVP, it was correct 40% of the time. And if you made the predictions of chemical shifts alone, it was only between 15 and 18%. And the next step was to connect this with sequential matching and use the sequential matching previously, which were given by line shapes, for an end-to-end assignment from a single HNA spectrum. And we thought the best idea was to integrate with CCPN. So using the CCPN backbone assignment module, given a single HNA spectrum, we could identify all the residues, label them I and I-1 positions, sequentially assign them with line shape matching. After sequentially assigning them to long chains, we could assign a long chains back to a primary sequence, all in CCPN as a plugin. So in here, you can see the example of the sequential matching. So in the backbone module, once you click on a residue, it would give potential sequential matches. And in here, you can see that sequential match to residue 71, 338 is a very good match with a correlation of 0.986. So it'd be quite a confident assignment. And this module was actually done by Harrison Wang from the Harvard team, which I then adapted to our per-root labeling. And afterwards, I had long chains for assignments for MVP, and I wanted to compare. So the sequence graph module in CCPN gives potential matches in the primary sequence for the long chains. So I did the assignments for the long chains with the chemical shifts and the neural network to see the comparison. And the number of potential matches for the primary sequence in sequence graph by the neural network reduced for some of the matches. So the very long matches, there was only 1 potential assignment. But if some of the long chains were 6 to 7 residues long, the number of potential matches reduced quite significantly, which made the assignment process easier. So in here, you can see the chain number 6. Chain number 6 had only 3 potential matches for the neural network, and there were 10 of the chemical shift predictions. And out of the 3 matches, the actual predictions by the neural network predict 4 out of 7 residues correct in the first place. And so with one HNA spectrum, inside CCPN, I could find all the I-1 residues, sequentially connect them, train the neural network, and assign them back to the primary sequence in about 3 to 4 days, which was much easier, much more automated, much quicker process than trying to assign it over the previous methods. And after long chains, trying to assign them to a primary sequence very quickly, in a couple of hours, just for the process of elimination, I could assign the chains, and with the neural network, I could assign 70% of the protein quite in just a couple of days. And if I use the chemical shifts alone, I could only assign 58% of it compared to 70% when we were using the neural networks. And I want to say thanks to my supervisor, Uluk Rasulov. It was his idea, and then the team at Harvard who helped me process the data and everything else with it.